All right, so now let's take a look at DNA replication. So again, James Watson and Francis Crick in 1953 came up with this whole DNA double helix thing. It should be something that you already know. But they also started to understand, what they figured out, that this structure provides a hint to how it's copied, and that is through replication, what we have called the process of replication. And then this whole first section of 11.2 is going through how there were various ideas as to how that occurred. There were 3 different main ideas that people looked at that made them think that perhaps was how it was done, and then there was a lot of experiments set up to try to test this. And then since they went through these various experiments to try to figure out which one it was, then they came upon this one model, the semi-conservative replication, which is where 2 strands of the double helix separate during DNA replication, and then each strand serves as a template from which the new complementary strand is copied. And then after replication, each double-stranded DNA includes 1 parental strand, also known as an old strand, so of the original piece, and then 1 new strand. So then there were these other models, as I mentioned, which were the conservative and dispersive. And this first section kind of goes through all of those experiments, which I don't expect you to know. You should already know that DNA is semi-conservative. Its DNA replication is semi-conservative. So we have, as you can see, here's our key here. Our blue ones are parent DNA. We take and we open this up here, break it apart, and then we go through replication on both strands here. And then we have an old strand, which would be the blue, the parent strand, and then we have a red strand, which is a new strand. And then, this is our first replication, so we have one that's the old and the new, and then if we go through more replication, then we have something that looks similar to this. So I don't expect you to know all of this, how they figured that out, or the other ones, so we're going to skip right to DNA replication in bacteria. So DNA replication in bacteria is similar in that we have this splitting apart of the DNA and then replication going in both directions, and we'll see that in just a moment. How it is different, of course, is that we're talking about a single circular chromosome rather than many chromosomes in a eukaryotic organism, for example, like humans. We can start here in the beginning. DNA replication has been well studied in bacteria because it's small and it's easy to do. It's harder to go through the process of looking at DNA in humans. We're big, we have lots of chromosomes, lots of base pairs. Making babies takes a lot longer for us than it does for E. coli, babies in quotes, because E. coli, of course, goes through binary fission, which isn't actually a baby but an exact copy. So we've used bacteria a lot to understand DNA and understand replication. So E. coli has about 4.6 million base pairs, and it's replicated in about 42 minutes. So interestingly, it takes about 1 second to add 1,000 nucleotides as it's going through replication, which is very, very fast. And it occurs with few errors, so it's very good at doing this. So DNA replication uses lots of different proteins and enzymes, and some of these may be familiar to you as we're going through this when you've learned it in other classes with eukaryotic organisms. So the first one that we'll talk about is DNA polymerase. So this should be familiar to you, DNA polymerase, also known as DNA Pol. In bacteria, there are 3 types of DNA polymerases. So we have DNA polymerase 1, DNA polymerase 2, and then DNA polymerase 3. And so what we found is that DNA polymerase 3 is the actual enzyme that's required for DNA synthesis, so going through replication. And that DNA polymerase 1 and DNA polymerase 2 are those that are required or used for repairing DNA that's been destroyed for some reason. So what we'll see here when we're talking about replication is this DNA polymerase 3. This is the one that adds deoxyribonucleotides to each complementary nucleotide on the template strand, one by one to the 3' hydroxyl group of the growing DNA chain. So this addition requires energy because we're growing something, we're going through an anabolic reaction. When we're taking small pieces like nucleotides and growing them into something bigger, it requires energy to be put into the reaction. So this energy is present in the bonds of 3 phosphate groups attached to each nucleotide. So here they show our guanosine triphosphate deoxyribonucleotide. So this is our G in our strand, and then these are the 3 phosphate groups. So the triphosphate deoxyribonucleotide. And so what we do is similar to what we do with ATP. They're similar to the ATP bonds, where these bonds right here are high energy bonds. We can go in and we can cut these bonds, cleave these bonds, and use that energy to take one nucleotide and attach it to another nucleotide. So when the bond between the phosphates is broken, diphosphate is released. So diphosphate, these two leave. We use that energy to then attach the rest of this molecule to the next molecule. So our OH group right here is our 3' end, and over here is our 5' end. And that's important when we look at replication. So then we have our formation of a covalent phosphodiester bond by dehydration synthesis between our incoming nucleotide and the free 3' OH group on the growing DNA strand. So our 3' right here will then attach to this 5' on another group. So we actually maintain this phosphate group here, and then it's going to be attached to this group over here on a different nucleotide, of course, not on itself. So, and then we'll see that in just a moment here. So let's talk about initiation. Remember, we have initiation, elongation, and termination. So the first piece here then is initiation, and this is initiation of replication. And this occurs at a specific nucleotide sequence, and we call that the origin of replication. So this origin of replication, of course, origin is where it starts. So that's where we start replication. In bacteria, we call it ORI-C. It's the origin, that's the ORI part, and then C is for chromosomal DNA. And we see a different origin, of course, for plasmid DNA. So our ORI-C is on its 1 chromosome. This is our origin. And so this is where we have our various proteins are going to bind and then begin this replication process. So the origin of replication is approximately 245 base pairs long and is rich in AT sequences. So there's a lot of adenine and thymine attached together in this one spot, high AT. So the proteins that we see that bind to it are important in making single-stranded regions of DNA accessible for replication. So we need to open up that DNA, remember, and get in there in order to replicate. So our chromosomal DNA is wrapped around histones in eukaryotes and archaea. So if you remember back to other classes when we were talking, or when you were talking about DNA, it's wrapped up very tightly around histones. They kind of look like little blocks of proteins where we take and we kind of crimp it, wrap it around like a thread on the histones, and then have another histone and wrap it around, and have another histone and wrap it around, which allows it to be very compact and take up little space in the cell. So we do see histones in eukaryotes as well as archaea, but then we have what are called histone-like proteins in bacteria. So they're not just like the histones in eukarya, but they're very similar. So it is supercoiled or extensively wrapped and twisted on itself to be in a small space. So that makes it very inaccessible. So it's very, very wrapped up, very tightly wound, so it makes it hard to get to. However, we have topoisomerase, specifically here, that are going to go in and change the shape of this chromosome. So they're going to go in and relax the chromosome. So for bacterial DNA replication to begin, we have to relax that by using topoisomerase 2. So we have topoisomerase 2 come in and unwrap and relax this supercoiling of the DNA. It's also called DNA gyrase. So then we have an enzyme come in that's called helicase, and then that's going to separate the DNA strands by breaking the hydrogen bonds between the base pairs. So remember, it's a double strand. We have our A's and T's and C's and G's matched up across the way from each other on opposite strands, and then they're held that way by hydrogen bonds along the interior of our DNA molecule. Those hydrogen bonds need to be broken and pulled open so that we can go ahead and replicate the DNA. So helicase does that for us. So topoisomerase is going to go in and kind of uncoil it, The way I think of it is like those super old telephones that had this curled up kind of cord to it that used to be attached to the wall. It goes in and it unwinds that curled up cord. It holds it so that it doesn't curl up again. And then Helicase can come in and it'll kind of unzip it. So Helicase comes in, it unzips these pieces, these hydrogen bonds, to pull it open so that we can access the different nucleotides. Then, so these enzymes require ATP, so it takes energy, ATP hydrolysis. As the DNA opens up, then we have Y-shaped structures called replication forks. So if you can imagine, let's see if I can draw right here, we've got our DNA strand, our double helix, and then if we go in, so first we've uncoiled it, which is why it's a little bit more straight here. Then we have Helicase come in and it's going to break these hydrogen bonds right in the middle. So then what we see is this opening up here. So it looks kind of like a Y. So we've opened up this space in here in order to get to these individual nucleotides in order to replicate. So we have what are called replication forks because they look kind of like forks. They're these Y shapes. And so there would be one on each end, right? So let me draw that again because we would have one on this side where it's opened up, and then we would have one on this side. So we see these replication forks or these kind of Y-shaped areas in the DNA. So we have two replication forks at this origin of replication, and this allows for bidirectional replication. So we can replicate on the top strand and we can replicate on the bottom strand at the same time. So then it looks like a bubble, right? Because we have the two Ys and then we see this little opened up section of the DNA, and we call that a replication bubble. And that's where we see the replication occurring. So the DNA near each replication fork, so where it's opened up at the junction there where there's still a piece together and then it opens up, is coated with single-stranded binding proteins. So that this prevents the single-stranded DNA from rewinding into a double helix. So I think of these single-stranded binding proteins, SSBP, our single-stranded binding proteins, as being kind of like tacks or pins that are holding it in place. So they attach to both ends or both sides, opposite sides of DNA, and they hold them there and make sure that they don't reform those hydrogen bonds so that we can get in there and do replication. So topoisomerase 2 in bacteria is going to go to the origin of replication, and it's going to relax the DNA from being all twisted up, from being supercoiled. Then Helicase is going to come in and unzip it by pulling the two strands apart. Then we have our single-stranded binding proteins, which are going to then hold that open for us. So then, now we have our DNA available. So once our single-stranded DNA is accessible at the origin of replication, then our DNA replication can begin. However, our DNA polymerase 3 is able to add nucleotides only in the 5' to 3' direction. So it can only be extended in this way. This is because DNA polymerase requires a free 3' hydroxyl group to which it can add nucleotides by forming that covalent phosphodiester bond that we talked about between that 3' end and the 5' end. So it's the 3' hydroxyl end that we saw on that G up above, and the 5' phosphate of the next nucleotide. So that means that we're going to have one strand, which is the leading strand, which can easily just have DNA polymerase 3 go in and start adding nucleotides because it has a free 3' end. But then we have the other strand, which is the backward strand, which is the lagging strand. And so we are not able to just go in and start adding to the 3' end because what's available is actually the 5' end. So it cannot add nucleotides if it doesn't have a free 3' end. So the way this problem is solved is with a small little piece of RNA. This little tiny piece of RNA, little RNA sequence, will be attached to that lagging strand, and it will provide a free 3' end. This allows the start of DNA synthesis. And so this is called a primer. So when our bacteria are going to go through the process of DNA synthesis or of replication, it has to have a primer available on its lagging strand. So we have this RNA sequence that's a primer. We use it just to get it going, and then it can come off. and then we have our free 3' end to get us started, and then our DNA polymerase 3 can add to that 3' end and continue elongating the DNA at that point. So the primer is 5 to 10 nucleotides long, and it's complementary to the parental or template DNA. It is synthesized by RNA primase, so RNA primase being the enzyme makes the primer, which is the molecule, and that is RNA polymerase. So unlike DNA polymerases, this RNA polymerase doesn't need the 3' group to make this molecule, so it can make it easily. We can make the primer, we can stick it on the DNA in order to get it going. So now that we have our 3' group, our DNA polymerase 3 can now extend the primer and add DNA nucleotides. So I want to go down to the illustration, and we'll go back to elongation. But so here is our illustration of a lot of what we just talked about. So up here is our general image here, our smaller image, not quite so blown up. This is our origin of replication, so our ORI-C. And then you can see that we have opened it up. You can see the replication bubble here. Looks like, similar to the drawing there. We've got a replication bubble. And then right here is one replication fork and one right here. And then we have our replication bubble. So when we blow up one of the replication forks, what we see here, again, is our topoisomerase, which is our enzyme. ACE is an enzyme that goes in and is going to uncoil that supercoiled DNA, kind of hold it flat so it's available. Then you can see here we have our helicase that we talked about. Our helicase is going to go in and unzip. So it has already broken the hydrogen groups, or rather the hydrogen bonds here. So it's gone in and it's broken apart those hydrogen bonds, so we've pulled the two strands apart. Then we have our single-stranded binding proteins. You can see here they're kind of anchoring it. Remember, I think of them as little tacks. They're holding them apart, making sure that they don't go back together. Then you can see here, this is our leading strand, and we have continuous synthesis here on the leading strand. So we have an available 3' hydroxyl group, and so DNA polymerase is immediately able to just add nucleotides as it goes along. And here's our DNA polymerase 3 that is doing that. So our 3' end is available, and it can add nucleotides as we are going this direction in replication. As our topoisomerase is going to be moving to the left, as it's shown here in the image, our helicase is also going to be moving to the left, continuously unzipping. Our single-stranded binding proteins are going to be moving to the left, holding that in place, as this, on our leading strand, DNA polymerase 3, is going to be continuously synthesizing DNA. Where we see the little bit of an issue is on our lagging strand. So here's our lagging strand here, the other one, the complementary strand. And so here you can see they've shown the RNA primer. So our little tiny chunk of RNA here, we have our RNA primase, which is the enzyme that has made that primer for us, is sticking this little chunk of nucleotides onto the DNA. And then you can see here is the DNA polymerase 3 that's able to utilize the free 3' end here to then start adding nucleotides this direction. So we're able to add nucleotides in this direction. You can see here's our free 3' end, and we can add nucleotides. But what we see, in order to have this, right, so we can add it in this direction. Actually, we'll talk about that in a moment in elongation. So those are the enzymes that we've talked about so far, where we have our leading strand and our lagging strand, and then we have to have our RNA primer in order to get that lagging strand going.